organizing committee members, Dr. Maheshwari Sab, for such a good opportunity to interact with the, forestry fraternity rather, right? And it's a very unique topic as always. It's an interesting topic to speak on forensics, and especially wildlife forensic, which is a bit a neglected field till date, even though after working in forensics for almost a decade, we know that okay, it's still underrated field compared to human forensics. But yes, let us do a bit to conserve the wildlife resources in such a way that the scientific applications are been enforced to the maximum possible extent, right? Sir has already introduced me and my university, so I'll not go deep into that. You know, Forensics is, I'll just straight away in a simple term, what forensics is. Investigation of any cases, samples, whether it is biological, chemical, or physical, where we need to use scientific techniques of basic science and the advanced instrumentation. That is what we do in forensics. So ultimately, when a crime is committed, so the major thing that we need to establish is to establish the triangle, the link between the suspect, the victim, and the scene of crime. Right? These three triangle, if we are able to link together, we will be in a position to solve the crimes in far more better way. Right. So as you can see on my slide, there are various techniques that are being used nowadays or since its inception, that means wildlife forensics, when it started all in the 90s, right? At Wildlife Institute of India, Dehradun. There are a few techniques that have been used so far. Of that, most of the cases can be solved using morphometric techniques. So there are various wildlife parts and products that we'll be discussing today. Which are the morphometric techniques that can be used for identification of wildlife part, may it be a whole part or may it be in the form of finished product, that we'll be discussing. The easiest way of identification and being, I guess, sir, can I intervene? If if I'm not wrong, most of the participants here are from the forestry background, or rather they are taxonomists, right?
Yes, yes,
Or an
yes,
academician.
yes,
Right. So yes, you all must be well aware about the taxonomy and morphometric identification. But apart from that, we have evolved over the few years now, or rather since last decade, since 2010 onwards, since the DNA technique got boom. Uh, People have been using the DNA-based examination for the wildlife parts and products. There are protein-based examination, and nowadays, a lot of scientists have moved on to the genomics. From genetics to genomics, people have moved. But one major thing that has been, lacking in the field of wildlife forensics that we have perceived over the years is the involvement of the human interface. Right. So we'll go through that one by one. Uh, there are famous cases that, or rather you can say infamous cases. The major difference that I'll be telling between the human forensics and the non-human forensics is, in human forensics, we are more so interested in establishing the individual identity of a particular specimen, right? Whereas in non-human forensics, as per the Wildlife Protection Act, you must be knowing Wildlife Protection Act 1972 of India. So, as per the law, we are just, required to establish the species identity from a given biological specimen, may it be skin, antler, horns, teeth, or you can say canine, whatever wildlife article it may be, or ivory. We need to identify the species, whether it is of lion, tiger, leopard, or whatever species it may be. So for that, for individual identification, we are in human forensics, we are using STR markers, that is short tandem repeats or nuclear markers. And since a lot of scientists are working on human forensics or human genetics, most of the protocols for identification of humans or biological samples originating from humans are validated and standardized. And you can find lot many experts in the field of human forensics compared to wildlife forensics. Now, talking about wildlife forensics, as I told, we are in requirement of establishing the species identity. So mostly for species identity, there are universal set of markers. We call it as mitochondrial markers like cytochrome B, 12S, 16S, or any other morphometric techniques that we can use. And for whenever a case is received, every time we, are required to analyze it and treat it as an independent case. Since a new challenge pops up in wildlife forensics, there are no standardized protocols, there are no standardized markers, right? And most importantly, a very less manpower is available right now in the country as well as worldwide. Very few experts are there in the country, right? So you know as already mentioned, wildlife forensics is all about identification of wildlife parts and product. May it be may the animal be killed by any technique, or you can say poached through trans-border smuggling, or while the animal parts or products are being sold illicitly in the open market, right? So over the past 2 to 3 years, we have noticed that the graph of wildlife crime has been escalated a lot. A lot number of cases has been seized. Why in India? What is that reason that a lot of cases are being seized from India, right? So obviously it is about meeting the requirement and the demand of the market. So India and Africa are two major continents where we are very rich in biodiversity. There are a lot of mega biodiversity zones, and a lot many flora and faunal species are there in India, right? So once there is a resources, obviously there is an exploitation of the resources. So the end users, obviously you must be knowing that there is a traditional system of medicine in China and other Asian countries. So we call it as traditional Chinese medicines. To meet the demand for the manufacturing of the traditional Chinese medicine, obviously China has been exploiting its natural resources. Now not so very natural, it's all artificial mostly in China, but a lot of natural resources has been exploited by China and its adjoining countries like Vietnam or maybe Thailand, or you can say in few parts of Myanmar or Bangladesh as well, right? So to meet that demand, wildlife products, parts and products like bones from carnivores like tiger or leopard are being smuggled back into the China through the porous borders. And we all know that there are porous borders all across the Indian subcontinent, right? So it is easy for the poachers to get the wildlife parts or products trafficked through such kind of porous borders, right? And there is as such no control over that. So we'll just touch upon few species that are in trade right now. Obviously, peafowl being the one of the very important species from the national perspective, since it is one of the flagship species. Apart from that, leopard, tiger have been given a lot of importance. We all know some of the herbivore species like black buck, or maybe some of the deer species, jackal, hare. All these species are traded. But then musk deer or ivory, that we all have heard about. But pangolins, I guess most of you might have heard about the name pangolin, right? So can anyone tell me? Rather, see, it will be more so a interactive session. That is what I would like. So I'll be asking few questions, and it will be kind of interaction. That will be what will be preferred from my side, if all agree, right? So can anyone tell which is the most trafficked species worldwide?
Yes, sir.
Yes. Yeah.
Pangolin.
Pangolin. Yeah, obviously. Right. So, people think that, okay, the poachers are targeting tigers a lot. But more than tiger, it's pangolin. The pangolins are being, killed for the want of scales that are being used in various traditional Chinese medicines. And it's probably estimated as per the reports that have been published by various, government and non-government organizations, that it's one of the species that may get wiped out even before the tiger or leopard, right? So pangolin is one of the world's most trafficked species, and we'll discuss a lot about how to identify the uh, pangolin scales, right? So, the major threat that we are facing nowadays, to our precious wildlife parts of wildlife, maybe flora or fauna, is major, obviously, is modernization, urbanization, industrialization, and that has led a lot of habitat fragmentation. Another key feature, obviously, is loss of genetic diversity. Since there is habitat fragmentation, the animal get a restricted land to, go through the routine activities for their dietary habit, for prey-predator relationship, or maybe to occupy the territory for the reproduction. And hence, it has led to the state of inbreeding, or you can say a lot many relatives have been, getting chance to breed with the closest one, and hence it has led to inbreeding, which may, harm the population in the longer run. And ultimate threat, obviously, is poaching, illegal killing of wildlife parts and products, ruthless killing, and its smuggling for various reasons. Now, why we need to control wildlife crime? It is obviously the fourth largest crime in the world. Can anyone tell which are the first three crimes, highly rated crimes in the world? Anyone?
Hello.
Yeah, can anyone tell
Illegal
the
arms,
first three? Yeah, obviously arms, narcotics, human
and
trafficking,
human trafficking.
right? These are the three highly rated crimes in the world. So every day in any newspaper, dailies, or maybe in the national or international newspapers, you must have read at least one news related to wildlife crime. Okay, this is where the poaching has happened or not. Why we need to control this? One major report that has been published by the international organizations is to execute first three crimes, right? The money that has been fetched from illegal smuggling or selling of wildlife parts and product, which is all black market. Obviously, the money has been utilized to execute the purchase of weapons, purchase of firearms. You must have heard about one of the infamous place, Ivory Coast, right in the African continent. So there, a lot of illegal activities are going on related to the drugs, human trafficking, or maybe for narcotics. Nigeria is obviously infamous for such kind of activities as well, that you must be knowing. Likewise, a lot many countries are there, that to just to name these countries a few. To control all these three activities, it is obviously necessary nowadays to control wildlife crime. So if wildlife crime is controlled, obviously people will get less chance. Rather, I cannot say that it will all be nullified, that controlling a wildlife crime will also reduce, or it will stop such kind of illegal activities like human trafficking or narcotics or XYZ kind of crimes. But yes, to a major extent, lot many things can be taken into control. You can see daily, a lot of news have been reported, maybe reptiles or mammals or any kind of poaching, right? So what happens in the real world scenario when you actually know the forensics or the nature of wildlife crime? In wildlife forensics, most of the crimes that have been committed are reported during the transit. That means when the wildlife article is being smuggled from one place to another place, may it be in train or may it be in the open market, at that time during the transit, the poachers or smugglers are being identified, right, and the articles are being seized. That means the actual scene of crime, from where the crime was committed, the animal was killed, where a potential chances of seizing the evidence is narrowed down. So if we know the exact scene of crime, where the crime was committed, where the can be gathered, and the same can be analyzed. Like nowadays everybody uses the smartphones, right? So mobile tower location can be one of the potential evidence. Fingerprints, obviously, which in none of the wildlife forensic cases, the evidence related to fingerprints of the perpetrator is being collected, which is a very, very bad state of investigation in India as far as wildlife forensics is concerned. In human forensics, it is a routine practice for the investigators. They by default collect the fingerprints of the perpetrator, if at all it is found at the scene of crime, right? So obviously, we need to identify the actual scene of crime. That is what the most important challenge is for us, right? And here, as you can see, to integrate wildlife science with the concept of the forensics, that means here by forensic we mean human forensics, right? And before that, obviously, I'll just give a brief of how it all started. Dr. S.P. Goyal, who is rightly considered to be the father or founder of wildlife forensics in India, has been, one of the commendable scientists that India has ever produced in the field of wildlife forensic. Many people might be aware about this magnanimous personality of Dr. Goyal, how he has contributed the wildlife forensic facility that has been set up at Wildlife Institute of India. The credit goes to Dr. Goyal sir and rather I was personally privileged to work under his able guidance, and what all today we are seeing is a collaborative effort of the efforts put in at Wildlife Institute of India and National Forensic Sciences University now. Apart from that, obviously, US is another country which has established a full-fledged wildlife forensic facility, right? So as I was talking earlier during my starting of my talk, it is important for us to form the linkage between the scene of crime, you can see here, the victim and the subject. If we are able to establish this and we get ample amount of evidence, we will be able to solve the crime in a better way than ever before, right? Of that, there are various enforcement agencies that will play important role in establishing this linkage. So one of the, as far as wildlife forensics is concerned, obviously the forest department plays very important role in detecting the crime, nabbing the poachers, and documentation of such kind of processes. Apart from that, nowadays custom has been very, very proactive, and many custom officers are now been trained dedicatedly in the field of wildlife forensics. That means many articles are being seized during the transit from India to the other borders, cross country smuggling. That means at the airport, at the checkpoint, the custom officials may come across the wildlife articles, and at that very moment, there are highly likely chances that the wildlife articles may be seized. So obviously the custom officers are nowadays trained in such a way that, okay, they will be in a position to establish the identity. We'll come to that, how, they go through the challenges. So one of the key feature of, the law that are there in India is, as per the Wildlife Protection Act, whether a person has killed an animal or not, but if that person, he or she, is found in the possession of the wildlife article, that is an offense, right? That is one of the salient feature of a Wildlife Protection Act. Another one very difficult task is, as I told earlier, most of the wildlife articles have been detected during trafficking or transportation. That means actual scene of crime is not known. Another important challenge is problem of sample identification. There are no standardized SOPs like what we have in human forensics. So every now and then a sample is seized, there is a new challenge that has been posed. Though many SOPs now has been standardized after publication of a lot of articles by researchers working in this particular domain, but still it's a very challenging task. One very, very important thing, to get maximum evidences from the scene of crime, we need to assess the crime scene in the most scientific manner. So crime scene management is very important, and one of the very sad state of wildlife forensic in India is 99.9% of the enforcement agencies does not follow the systematic SOPs for the crime scene management. It is almost parallelly similar, or rather similar to what we follow in human forensics, right? And I've been very much privileged that, okay, I have got the opportunity to work in both human forensics and wildlife forensics, and hence I know how to, execute the SOPs that are there for, non-humans as well, the one that we follow in human forensics. So it is easier for us to, execute for both wildlife forensics, and that's the place where, we are strong in terms of wildlife forensics here in NFSU. Here, I don't know if there are any veterinarians or doctors or pathologists here, but the way sample has been collected, most importantly after collection, the way it has been preserved, packed and forwarded is very important. Again, this is a very, very separate topic. I need at least 1.5 hours to speak on the sample collection, preservation, packing and forwarding of the samples. So we'll not discuss that, but the way samples have been preserved is very non-scientific, rather, people use formalin, right? So how many of you agree that formalin is a better preservative for storage of samples, biological samples? I guess most of you agree, right? Can anyone just tell me, sir? Yeah, are you all there?
Yes, sir.
Right. So as far as DNA forensics is concerned, where a sample has to be subjected for DNA examination, preserving a sample in formalin is like a sin. It is itself a crime. Once you use formalin for preservation of wildlife samples that are meant for DNA examination, that means you are destroying the DNA itself. So it is better to use formalin. It is better to use silica gel or maybe alcohol. Yes, please.
You have a question?
Yes, please, sir.
Dr. Yumnam, you have a question here? Please carry on, sir. I think she mistakenly unmuted herself. Yeah.
Okay, fine. Right. Another important thing in forensics, it is very important to, maintain the chain of custody of whatever article, right from the scene of crime, whatever article we have collected as an enforcement agency, till its final proceeding in the court, who has collected, how it has been forwarded from one agency to another agency, the exact detail has to be there. There should not be any tampering. Unfortunately, in most of the wildlife forensic cases, because of lack of awareness amongst the enforcement agencies, yes, this is not followed the way it has to be, right? Anyway, these are the challenges that we are facing. Now coming to the laws, obviously Wildlife Protection Act is a major law, but apart from that, when an article is booked, most of the enforcement agency book the perpetrator under just one law, that is Wildlife Protection Act 1972, its various sections and subsections. But there are various other laws that will also help us in better conservation of wildlife species and controlling the illegal trafficking. Biodiversity Act 2002 obviously is one of the very important act where our valuable resources can be preserved. Apart from that, since human is involved always, so whenever there is a human interface, we don't impose sections of human related crimes, that is Indian Penal Code or CRPC or maybe other domestic acts, right? So every time a human is caught as a perpetrator or a suspect, you should always, if at all applicable, as and when applicable, Indian Penal Code sections or CRPC, wherever it is applicable, needs to be imposed and included in the FIR or POR, you can say. So yeah, now coming to the exact topic. So you must be knowing various methods of poaching. So if you know how the animal has been killed, tentative ways the animal has been killed, you may find, you are highly likely the probable person to find the related evidences, right? So coming to first method is snare wire, obviously, where cables are placed around the trees, and the animal, once it walks through, the animal gets chopped up, right? So once, while placing the snare, the perpetrator will be leaving a lot of evidences, foot marks, fingerprints, right? and everybody uses the digital devices. So obviously the mobile tower location, though in forest area, obviously it is difficult to trace the signals of the mobile tower mobiles as well, but still it is one of the viable evidence that everybody can look for. Snare wires, obviously for the majestic species, a lot of times snare wires are there, though not that lethal compared to not that lethal for the majestic species like rhino or maybe elephant, but still can be a potent tool for killing the animal. Bird trapping is one of the common practices that has been followed extensively in India. So that is one of the technique where the animals or birds are being killed. So I'm not going deep into the theoretical aspect of different methods of poaching, just the overview how it is there. This is very important. Lag traps, like suppose tigers or lion or maybe snow leopard, if at all, which is less cited, or the cases are less reported for the lag trapping of the snow leopard, but a few majestic species, leg traps are used, right? So as soon as the animal walks through, the animal gets the leg of that animal, paw of that animal gets trapped over it after it stumbles upon it, and then it gets entangled, and subsequently after trapping, there is a huge roar. So one question arises: uh, the officers patrolling in that vicinity, right, may hear the roar of the majestic species. So you would be wondering how could the patrolling officers, maybe forest officers, don't get a idea of the such fishy roar of the animal in the forest area if the illegal activities are going on, right? So what they do is, as soon as the animal gets trapped on this kind of leg traps, they first poachers, what they do is they just break the vocal cord of the animal, right? So once the vocal cord is broken, that the huge roar that would have been heard for kilometers in the silent zone of the forest will not be heard, and the activities may go unnoticed, right? That is what the common modus operandi is by most of the poachers, right? Yeah. So this is one of the case study that has been there. So what they do is they make the animal to follow one path, and on that path they might have kept the this lag traps or peat holes under which the animal gets trapped, and subsequently they break the vocal cord and execute the act of killing or poaching, right? So these are various methods that are being used. So we'll not go deep. Again, these are all the case studies that would not be interested to discuss right now, keeping in mind the time constraint. Spears, obviously, one of the traditional technique that has been used, again, a lethal weapon for the close encounter with the animals. Mostly, the domestic species are being killed. Apart from that, few smaller carnivores or small mammals are being killed using the spears. And they again break the vocal cord of the carnivores to stop the huge roar of the animal, as you can see on the slide as well. So weapons can be any type of weapons, and you can see why this could be one of the reason why the animals are killed. One of the reason, intrusion in the human landscape. Rather, we have intruded in the animals' territory, but you can say the animals, once they intrude the agricultural crop, or maybe nearby the vicinity where they come to feed on the livestock or cattle, the people living nearby the area try to kill that animal, right? Sometimes weapons are being used, or sometimes guns are being also used. This is very interesting. If the animal is found, the skin of the animals is being sold in the market, where the skin needs to be, if the skin is intact, that will fetch a higher price. I guess all of you agree, right? So if there is any cut mark on the skin, the amount gets reduced for selling that skin. It will be difficult, so everybody would be willing to get the finished product, right? So what people have done is, this is one of the classical case of the otters, where we received some of the skins of otters, and there was no signs of killing, no weapons were found. So we were wondering that how could this happen, that there is no sign? How could these otters be killed? There was no bullet hole entry or exit wound. There was no tearing mark of the sharp weapon being used. Nothing. So we were just wondering. So just out of curiosity, we just asked the enforcement agency who had sent the uh, seized consignment to Wildlife Ministry of India, and we found that okay, such kind of seeds of strychnine or nux vomica, you can say the poisonous seed. You can also say arrow poisons. Just they put the cut mark over this, and then they just release it into the water body, and otters have been inhabiting nearby the water body, right? So as such, all the otters would die. Okay. So this could be one of the reason. Poisoning is one of the technique, and how to identify whether it has been poisoned or not. Obviously, you can look for the evidences related to the poisonous seeds or any other such kind of material, as well as the postmortem examination would confirm the same. But to go for postmortem of such kind of huge consignment, it's difficult, and hence, as per the Wildlife Protection Act, we just need to identify that the killed species or specimens are of which species. That is what we are restricting up to. Okay, electrocution is one of the method of killing firearms. One of the problem here, okay, just for your kind information, audience, that here at National Forensic Sciences University, we have the state-of-art facility for examination of firearms. We have ballistic testing center, bulletproof armored materials are being tested here. But here or elsewhere in India or globe, there is no facility for the identification and examination of the non-standardized firearm. You must have heard about the kattas or country-made firearms, right? So in wildlife forensics, mostly whenever the crime is committed, the poachers mostly use the country-made firearms, and hence it is difficult to identify such kind of firearms. We need the reference standard, and it can be fired in any direction. You must be knowing that, right? You can see such kind of modified firearms are being used. So it is difficult to go for the ballistic examination. That is again a challenge, and someone can come up with that as well. Okay. Yeah. So these are most common evidences that are being seized. Mostly, the tissue samples are being forwarded to the best of my knowledge, since based on the experience that we have gone through over the years. Tissue samples, ivories, claw, sometimes hairs, bare biles, paint brushes are being seized nowadays in numbers, and any other finished leather products are being seized, right? So how to identify? Just a quick view. First half of my lecture is over, I guess. Sir, I guess we have 25 more minutes to go.
No problem, sir. We are not, putting our clock restrict your valuable knowledge for us. So please.
Oh, thank you very much, sir.
Feel free.
Sure, sir. Yeah. So, for species identification, as I told earlier, it is very important to identify the species from any given biological sample that has been seized in wildlife forensics. So, four major domains are being, explored nowadays. Of that, two are the most common that everybody must be knowing. That is morphometric examination, whereby we use conventional methods. So there are a lot of experts here. Maybe I presume there are a lot of experts in the session itself right now who must be knowing about the pug marks. Then we have M stripes, a very flagship project that has been, undertaken by WI, ZSL, or WWF India, right? So it's a collaborative effort of a lot of organizations. M stripe has been a very good success, just based on the photo of the camera trap that is right now restricted to tigers only, but maybe extrapolated to the other species as well, like leopard. Based on the rosette pattern or the stripe pattern, you can identify the individual, whether it is originated from a particular Corbett tiger reserve or Panna tiger reserve, or likewise any other protected area. So apart from that, these are all conventional techniques. The easiest being microscopic examination of the wildlife parts and products. Suppose you have hair sample. So simply based on the cuticular examination or medulla examination, it is easy. I will go through that. Apart from that, nowadays we have explored anthropology and odontological examination of the wildlife parts and products, where if you have skull in your hand, you need to identify whether it's a tiger skull or a leopard skull based on the morphometric features. It is now easy compared to earlier, where but still it requires a lot of expertise, where you would be in the position to differentiate just based on the sutures, opening and closing of sutures, the fissure marks or the apertures of the eye, whether it is a specimen of tiger, leopard, or any other species. It requires a lot of huge database. Still, it is under preparation. A lot many scientists have been working on it, including our team here as well. But still, it's a huge task. So someone can take up this task as well. The problem is obviously to generate a database, we need samples, and it's difficult to get the samples of skull from the enforcement agencies for various animals. One very modern technique that we are endorsing and we are being routinely using is, imaging techniques like X-rays and CT scans, right? And you just need to use the X-ray to, highlight the inherent features of every article that will go through that, right? And one most important technique that people have been using is molecular technique that includes DNA-based examination, mostly mitochondrial and microsatellite examination. For a few of the samples like venom, if suppose there is a venom, it's difficult to extract the DNA out of venom where multiple species have been involved. So for that, protein-based examination is endorsed, and yes, very few scientists are there in India who can actually establish the identity of a snake species or any other venoms, any other species based on the mixture of venoms. Serological examination is not needed right now, since for non-human forensics, it is not that explored and important. Yes, analytical technique. So for wildlife articles that are in finished form, where it is difficult to extract the DNA out of it, the morphometric features has been lost. Like for suppose ivory, if it is in powder form, it's difficult to identify, or maybe rhino horn, if it is in powdered form, meant directly for the traditional Chinese medicine preparations, it is difficult to establish the identity, whether it is a ivory product or maybe rhino horn or maybe any other articles. For that, analytical technique like gas chromatography, mass spectroscopy, HPLC, FTIR or FT-Raman, likewise many other techniques are being used. So yes, we are privileged to have such kind of in-house techniques here at National Forensic Sciences University, and yes, we are exploring such kind of techniques a lot. Except NFSU, there are hardly any institutes. I don't see any other institute, or I haven't come across any institute where they are being using analytical techniques for identification of wildlife parts and products, right, in a routine forensic cases. And this will obviously be there in the future radioisotope mass spectroscopy, and that can help us a lot. Apart from that, now at the scene of crime in human forensics, we are using 3D scanners, and drone surveillance obviously could control a lot of illicit wildlife crime activities, but still it's way to go before it is actually executed. So coming to I'll just briefly go through some of the techniques we have gone through this. Yes, every animal has a unique stripe on their body. So obviously a commendable work has been done by obviously the agencies that has been named here, and nowadays tiger stripes, if we have the database of the stripe pattern of tiger, we can exactly say that this particular tiger originated from Corbett, or this originated from Panna, or likewise, right? Since like fingerprints, the stripe patterns are also unique. For those who are really interested to start their career as an academician or a research scientist in wildlife forensics, the easiest way to start is obviously by taking up a work related to morphometric examination and microscopic examination. So Wildlife Institute of India, Dehradun, obviously has published one of the very good manual whereby you can use the key characteristics like cuticular, medulla, and cross section for for the identification of various items, maybe paintbrush, maybe the skins that has been seized, or maybe any other wildlife article made in finished form or maybe in a raw form. So there are 53 mammalian species for which the characteristic has been defined in this manual. You can get it online. If at all it is not available, if you are not able to find, I'll be happy to share this manual as well. What is there in this manual? Identification characteristic of 53 species that have been listed in this manual based on cuticular pattern, medullary pattern, and cross section. But cross section is not that important. We have used SAM as well, scanning electron microscope. Right. You can see here, these are all cuticular patterns that are unique to a lot of group of species. Medullary pattern obviously is unique for a lot of species, and obviously we are also working on the atomic force microscopy, which is obviously the thing that will be there in future. You know, is rather at a nano level atomic force. That means, you know, at a nano level we are identifying the species for the hair, and identification of species based on, you know, such kind of characteristic at the nanotechnology, nanoscales, right, rather at the micro scales. I guess you might have heard about shawls, right? So there are a few species like shatus. We also call it scientifically as Tibetan antelope, which lives in the Changtang Reserve. At minus 40 degrees centigrade, this more than minus 40 degrees centigrade, the species can survive, right? So salts that are prepared from this animal are being traded illegally, specifically in the China and the Tibetan plateau, right? Changtang Reserve, where, you know, we just use the guard hairs. You can see here, these are the guard hair. You just need a stereo microscope and the compound microscope like this, comparison microscope. Compare the cuticular pattern of the species, and you can identify whether the shawl is been prepared from any domestic species like pashmina, or maybe a domestic sheep or yak or any other species, or it is from the scheduled one species, that is shatus, right? Apart from that, obviously any morphometric characters study can be, you know, complemented obviously with a more robust technique, that is DNA based examination. And it is always necessary to trace the origin of wildlife parts and products. So geographic origin of the parts and product can be done using the haplotype database, and identifying the haplogroup from where, you know, it will help us in identifying whether the unknown specimen has been originated from which part of the country or which part of the world, provided we have the reference database for other populations as well, right? Coming to another flagship species, as always there, tiger. Everybody knows tiger, and you can identify whether the given specimen is of tiger or not, I guess rightly so, right? Audience, should I take a pause, or it's a continuous talk?
It's a continuous
Are
talk.
you all getting?
Of
Yeah,
course,
you are
sir.
getting or
Please.
not? Should I?
Continue. Very interested, sir.
Fine. So, okay. So each and every part of tiger has been traded. You know, as you know, people say, "Jitna noch lo tiger se," na? Kis insan se. So people have been, you know, exploring and rather exploiting tigers a lot, even the leopards as well. So you take tiger skin, bones, eyeballs, brain, tail, heart. Every part is been traded for their demand in traditional Chinese medicine or as a trophy material, right? So how to identify whether it's a tiger or not? Like, okay, as you can see here, there can be various possibilities and scenarios of death, like whether it has been accidentally killed or whether it has been killed by poisoning. And in human forensics, we are more interested in time since death as well, when the, you know, death occurred. But unlike that, in non-human, we are not interested in when the potential death occurred, except in few cases, right? I guess everybody can see this photograph. What it is?
Yes, sir. Yes.
Right? I guess it is tiger, right? Everybody can see it's tiger.
It's a false pelt.
Okay, it's a fake skin, right? So how to identify? Everybody have seen tiger, and if you might have observed, the stripe pattern in tigers are asymmetrical in a natural population. Whereas here you can see the stripe patterns are almost symmetrical. Someone has painted that with a dye material. Okay, I have the video of this kind of skin as well, how they prepare, right? Using the turmeric and other chemicals they treat and then just paste with the dye. So one of the easiest way is to identify this fake and the genuine tiger is you simply plug the few hairs from the mid dorsal part, dip it either in the nail polish remover or maybe in the alcohol that is readily available around your places. And if at all that natural color is not there, if at all artificial color is there, it will, you know, vanish. It will wash out. That is the easiest way to identify the genuine in the fake skin. Another one obviously is to identify the species using morphometric characteristics like cuticular and medulla that has been defined in that manual. So I'll not go deep into that. Right. Likewise, you know, same for the leopard as well, whether it is fabricated fur coat or maybe it's a genuine one. The rosette pattern, you know, says it all. Like stripe patterns are there in tigers, there's a rosette pattern in leopards, and even the dye blobs have been used to, you know, prepare such kind of artificially looking leopard fur coats or any other material thereof. But it can either be genuine or it can be fake. So one of the challenge in wildlife forensic is to identify whether it's a genuine wildlife part of product or it's a imitated one or it's a altogether fake one. Yeah. There's another manual been published by Wildlife Institute of India. A few scientists who have been working. Dr. Kumar has been very active in morphometric identification of wildlife parts and products, and you can find the identification characteristics of the canines, the teeth, or any other carnassial patterns of various species. Right. So how do we identify species from teeth or maybe bone? So looking into the shape and structure of either canine or claw, skulls, or the size of the bones, the stature of the bones, there is one specific formula like what we use in human forensics, the Gustafson's formula to estimate the age of the person. Likewise, in non-human forensics, we identify one of the inherent characteristic that develops and evolves over the course of time. We can identify the age, we can identify the species, and to some extent, not 100%, to some extent, the individual identity of the animals as well. Right, you can see here the sagittal crest, the specific peculiar pattern of sagittal crest will be different both for the leopard as well as tiger. Always when in wildlife forensics, the most common confusion whether it is a skull of a tiger or a leopard. So it is easy to identify based on the eye apertures, the sagittal crest, you know, the nasal apertures. Likewise, all these characteristics together we form into one morphological sketch or graph, and then we identify whether it is a tiger skull or a leopard skull. Likewise, the other bones are of tiger, leopard, a domestic species, or herbivore species. Likewise, so what we have explored is, you know, X-ray can be a very important tool in identification of bones as well. If it would have been fake like this, you can see here this is all opaque. Likewise, here there is a deposition of the cartilaginous material or the soft tissues as well sometimes, and that can give a specific pattern for a specific species. So we are also trying to come out with a manual related to the X-ray examination of the different species, where, you know, a tiger bone, femur of a tiger can be differentiated from that femur of the leopard, or from that of the snow leopard, sloth bear, or any other species, right? The selected species, whatever the specimens that we have right now. Right. But then we have moved a step ahead. Suppose we have one specimen of any skull. What we do is we take that photograph, we project it in the software, and we will just try to prepare the 3D sketch or the 3D prints of the skull. Likewise, you can see here, that will give you an exact idea of the tissue deposition or the muscle depth, right? And can give you the identity whether it is a tiger, the skull of tiger or leopard or any other species. Right now, the most common feature that has been explored, as I told earlier, is zygomatic arc that has been explored to identify the species or differentiate the species. You can see here these are all fake articles, right? So it does not give any characters. It's all looking opaque. Likewise here as well. Whereas this cartilaginous characteristic of the antler or maybe for ivory, it all is well projected. Okay. Likewise for canine, one of the important characteristic of the canine of cat family is the presence of this groove, minor groove, just at the interior end of the starting of the claw, and it is present in tiger, leopard, as well as other cat species or felid species. Whereas if you see the canine of the domestic species, this kind of groove will be absent. Right, so this is the part that remains inside the gum area, and this protrudes outside. And since the carnivores has the typical habit of tearing the fleshes of their prey species, it is somewhat wear and tear will be found here. And depending upon the dietary habit, the canine may be blunt, sharp, or maybe chiseled. Likewise, like hyena, if you see hyena or wolf, they are all scavengers, and hence mostly it will be blunt. Likewise for the pig as well, almost a triangular shape. You can see another characteristic of the claw, right? Claw, people use claw for as a superstitious belief that okay, wearing the pendants prepared from claw or canine gives strength, right? So in that case is the inherent characteristic, whether it is a genuine claw or not, can be identified. See, one thing, as I told earlier, is there are inherent characteristics that we need to explore, right? So if it is a fake claw, you can just get it polished from the buffalo horn or so, but it will not have the inherent pattern like what you see under the X-ray. You cannot change this pattern. You cannot make this pattern in the fake claw, whereas this is the characteristic that is found only in the genuine claw. So that is easiest way to differentiate whether it is a claw of tiger, leopard, or it's a fake claw. You can see here, and this bony part will always be fitted outside the genuine claw, and this claw part is always inside. It's all keratinous material, and hence the keratin deposit pattern is unique for all the species, maybe tiger, leopard, or any other species. Likewise, there are a few more things to discuss, but keeping the things time constraint in mind, I'll just come to a few more important points. uh, you know elephant are being illegally poached for mostly ivory. So how to identify this is genuine ivory or not? So there will be Schrager lines, crisscross lines that are will found. If you take the ivory and cut the cross section of that ivory, polish it with the buffing material, you will find the presence of Schrager angles and Schrager lines, right? Which is found only and only in the elephants, or you can say ivory, right? So if you find such kind of crisscross lines in a specific pattern, you will say that okay, this is a genuine one. And apart from that, the fabric imitated articles are also there. You can see such kind of seized material was also sent to Wildlife Institute of India earlier, and inside that, when we broke open this material, we found that it's all white cement, and over that the resinous material has been uh, pasted, right? So these are all fake items as well that can be found. um, Rhino horn, obviously, rhino horn is a tuft of hair fused together, right? As simple as that. So, if one on rhino is there, a rhino horn is found. Since it is a tuft of hair fused together, if we see the base of the rhino horn here, suppose this is a rhino horn. If we see the base here under a microscope, we will find the presence of cellular pores, right? So suppose this is rhino horn. We took the section of it. We found under the microscope that okay, there are cellular pores, emerging out of the base of the rhino horn, which will not be present in the fake one. You can see here, this is all fake. This is all fake. This is all fake rhino horn, and this is a genuine rhino horn where we found the cellular pores. So that is one of the easiest way to identify whether it's a genuine rhino horn or not. Basically, it's a keratinous material, a tuft of hair fused together. Right. Likewise, yeah, there are two species of pangolins that are found. So, researchers working at Zoological Survey of India as well as Wildlife Institute of India, they have already developed the key for the identification of the pangolin scales, whether the scales belongs to Indian pangolin or the Chinese pangolin, both based on the morphometric characteristics as well as that of the DNA based characteristics. So they are also developing the pangolin identification system like we have in CODIS system. It's very important to identify the species, whether it's an Indian pangolin or a Chinese pangolin. Sir, I'll just take 10 more minutes with your due permission, if you allow.
No problem, sir. No problem, sir. Please.
Yeah.
We are
I
thoroughly
hope it is.
enjoying.
Okay, fine. Fine, sir. Thank you, sir. So one thing, very important thing to discuss here is coming to the actual talk now. Just a 10-minute of, eye-opening session. One of the classical case that ever happened in the field of wildlife forensics, as far as investigation is concerned, is the Gir lion poaching case and its subsequent investigation. Probably that is the only case so far happened in India which was investigated by the dedicated forensic experts, police, and close coordination of the entire administration, where 8 to 9 lions were killed, and it was a challenge for the investigators who were the poachers and how to identify them. We made the best. I was in the heyday. I just started my career in forensic science, and I got this opportunity, and we're glad to be part of that investigation, obviously led by one of the premier IPS officers of the country. In this case, all the evidences related to human forensics were gathered, may it be fingerprint, may it be footprint, may it be mobiles, may it be the physical evidences, vehicles, etc. So all the evidences related to human forensics rather than wildlife forensics were covered. All the evidences were matched, and subsequently the suspects were caught, identified, and later on, there is one technique of interrogation, we call it as narco test. You must have heard about narco test and lie detector test. So all the suspects were subjected to narco test, and based on the firm investigation of human forensics and wildlife forensic interface, this case was solved. And today, based on that investigation, all the 28 convicts have been behind the bar. So can you imagine one of the very important clues that turned the table round? You would be surprised to know it was the matchbox that was used and seized at the scene of crime, and the wrapper of salt and dry fruit can be a very potential evidence, in narrowing down the field of investigation. That matchbox belonged to one of the particular place in Madhya Pradesh. This happened in Gujarat, and based on that, it was found that okay, this type of matchbox and this type of salt wrapper is found only in Madhya Pradesh. And based on that, the entire investigation took a turn, and later on the routes were investigated, and we found the suspects. And yes, it's a successful investigation that has been carried forward. So you can just imagine the importance of human interface. Like I told, the technological advances plays a very important role. Once we have technology, why not to utilize that, right? So this is another classical example of what we use in human forensics. We always neglect the human related evidences, right? Suppose you are at the scene of crime, you have the liberty. Yeah, this in wildlife forensics is a bit not that feasible right now, except the cases where we know the actual scene of crime, the major cases or the major seizures that we find. But nowadays we are using the laser scanning technology. Suppose the enforcement agency has seized few articles. The case has been forwarded to the forensic laboratory. A report may come 6 months, 5 months, or 3 months, or 1 month later on, after the due submission of the evidence samples. The case report submitted in the court, since there is a huge pendency in the court, it may come in trial 2 to 3 years, or 4 years, or 5 years later on. So what were the sequence of event that happened 4 or 5 years back? Nobody has that very sharp memory to remember each and every bits and pieces of the information that was gathered at the time of actual scene of crime. So it is always necessary to reconstruct the scene of crime and document the scene of crime in the most scientific manner and using the advanced tools and techniques. So nowadays we have started using this laser scanning technology. We call it as 3D crime scene scanner. And what we need to do is just like a camera. This will rotate 360 degree. You just need to place this 3D scanner here in the middle of the crime scene. It will rotate it 360 degree and scan the entire scene of crime, give a sketch like this, 3D sketch, right? That will help us to know the exact dimension of the object, evidences, where it was placed and what was the distance of it, and we will be able to successfully defend our case in the court of law. These are all the questions that are asked while defending the case in court. That okay, where did you collect the evidence? When was the evidence collected? How far was the evidence from that particular location or the dead body of the animal? Likewise, many questions can be asked by the lawyers during the court trials, right? So if you are not in a position to answer a few questions, few important questions, you may lose out the case. So yes, using this 3D crime scene scanner and latest technology, it is always useful for investigation of the cases in a better way. Another important contribution that we made and that will be useful for the wildlife forensics and the human forensic fraternity is we are introducing mobile forensic investigation van across all the districts of India. A very flagship project under the Ministry of Home Affairs, where all the districts will be provided with the forensic investigation van designed by obviously National Forensic Sciences University. And these kind of vans, and another version of the miniature van has all the kits that can help us in on-spot identification of wildlife parts and products. So if you have any wildlife part or products or maybe human interface, on-spot identification is very important. Half of the case will be solved on-spot only. Right? And you can see this is the vision that has been led by our Honorable Vice Chancellor Dr. J.M. Vyasal, who has been recently conferred with Padma Shri yesterday itself. Right. So the vision that has been carried forward, it will be very important. Again, many laboratory processes are time consuming, right? So what has happened is there is one technology, we call it as rapid HIT technology, where earlier from this sample till extraction of the DNA till sequencing, it was taking almost 2 days per sample, rather, or a batch of samples to get the report. And that too, with a huge pendency, it takes long time, and then subsequently we analyze the data. Now this all has been clubbed in one, and this being a very mobile instrument can be carried in the mobile forensic van. Just simply input your biological sample, and within 45 or 50 minutes you will get the sequences or the report, DNA related report of this any particular biological sample. So that is what the technology plays a very important role. Another technological advancement that you always need to look for and use is obviously we all live in the digital era, and we have been becoming the slave of the mobile computers, laptops, and many digital gadgets. So computer forensics plays a very important role. Mobile forensics plays a very important role. Though this is not my domain, I'm not the appropriate person to speak on this mobile forensics or network forensics or computer forensics. But yes, as an investigator, you should be aware of what are the tools and techniques that can be used and that can come to your rescue as and when needed. And people should make best use of the mobile forensics and its related data in and out of the mobile towers, how many active tower locations were there, if there is a potential attack, cyber attack, how can that can be dealt. And obviously this is not the domain that everybody can work on, so we need a dedicated team of cyber forensic experts, and they would be the best person to investigate such kind of cases. So obviously when you visit the scene of crime, there are a pool of experts that can be taken to the scene of crime, and everybody can contribute and make the case a very successful story and give a better justice to the victim. Here in our case, obviously, non-humans, right? So I guess that is it from my side, sir. Is there anyone who is having any question?
Thank you, sir. Thank you very much. That was really an illustrative and a holistic approach you have provided us of the entire spectrum of the advancements in the wildlife forensics. Of course, you have taken it beyond, the wildlife as well as the human forensics, and that was really interesting while listening your lecture on this very important topic. So now the session is open for everyone to please ask your questions to Dr. Malay.
Sure, sir. I am open for the.
Dr. Malay?
Yes, sir.
Hi, I am Adil from Navsari Agricultural University.
Yeah, please.
I do not have a question, but I have compliments for your wonderful presentation.
Thank you, sir.
We have not met, but we are working with Dr. Ravtani.
Oh, good.
Along with many projects, a recent project on Snake Research Institute, which is being established at Dharampur.
Yes, Dharampur. Yeah.
But I'm really happy to see you, and it was really a very nice presentation. Looking forward to working with you as well.
Sure, definitely, sir. We are also analyzing the mixture of snake venoms in our laboratory. We have a state-of-art laboratory here.
Yes, I visited.
And I know Dr. Ravtani as well working on this, and recently submitted one of the proposal on that.
Yes. So it was wonderful presentation.
Great.
Thank you for sharing such a.
Thank you, sir. Yes,
Great.
sir. I would love to have some more
questions and compliments. Sorry. So there is a question that how similar and different the flora, fauna, and human crime investigation techniques in forensic science.
Okay, sir. See, as I told, for human forensics, it's one straight line of action where standard SOPs are been designed. For non-human forensics, if we talk about fauna, we have discussed a lot on fauna. Like we have any biological sample from non-human origin, specifically animals, parts and products. We just need morphometric examination, as I discussed. If not possible through morphometric examination, identify the species using DNA-based technique. And if not possible through DNA-based technique, yes, we have another line of parallel action, that is analytical techniques for the finished products. Whereas for floral species, mostly you must have heard about the red sandalwoods, right? That has been illegally smuggled, right? Or maybe sometimes there are cases of disputes of geographic identity. A story, Rasgulla kahan se aaya, Odisha se ya West Bengal se. Likewise, there are various plant species of mango, obviously, whether it is a variety from Alfonso from Nagpur, or maybe any other country, Panjgini or so, right? So in such cases, we need to identify the origin, and in few of those cases, yes, it's difficult to identify the origin, but the line of action remains same. Yes, we can go for molecular analysis for plants as well. But for plants, we need a dedicated molecular markers. I guess I have justified the answer. If not, please ask.
Yeah, of course. So since everyone is talking about the recent movie, Red Sandalwood, Pushpa, the first part, The Rising. So if we have like molecular markers, particularly for the red sandalwood, they are coming from Tamil Nadu or some other parts, and wherever in the world they are seized, so we can actually identify the origin location where they have actually originated, right?
Yes, yes. Geographic origin of articles is necessary, and provided we have a database, DNA database of those particular places, the haplogroups or haplotypes, yes, it is possible. Though, unfortunately, because of time constraint, I was not able to show the that haplogroup and haplotype analysis of the species, but yes, it is possible, sir. But we need a comprehensive sampling, the database, and then we can identify.
Sure. Hope Pushpa is not listening our talk.
Sir, unfortunately,
Anyway,
I
so
haven't
that
seen
was,
the movie.
you must watch it. That was a really nice movie. We have recently watched it, just
Sure, sir.
a couple of weeks back. So anyway, coming to the main business, it was really interactive and enlightening, talk for all of us.
Thank you, sir.
And we look forward to your similar kind of cooperation and guidance for future academic programs. But for a formal vote of thanks, I would now request my colleague Dr. Kaushal Singh to kindly deliver the vote of thanks to you.
Thank you, Dr. Maheshwari, to giving me this opportunity to give my sincere thanks to Dr. Malay Shukla. Good evening, Dr. Malay Shukla, for giving your crucial or important time for sharing your valuable talk with our participants and enriching our participants as well as us with your vast experience in wildlife forensics and various modern tools and techniques which are using for the investigation of the wildlife crime. So on behalf of my organizing committee and my own behalf, I would like to thank you very much for giving your valuable talk. Thank you, sir. Namaskar.
Thank you very much, sir. Sir, even I'd just like to add a few points. I would also like to thank the entire organizing committee, specifically Singh Sahab, Dr. Maheshwari sir, for being in continuous touch with me and giving me an important opportunity to interact with the participants here and delivering such important talk. Right. Hope to carry forward this collaboration in future, sir. Thank you very much, sir.
Thank you, sir. Thank you.
With your due permission,
that
sir.
Was a, yeah, sure, sir, sure. I know. We know that you might be busy. Sure, sir. You may sign out, sir. You may sign off.
Thank you, sir. Thank you, sir.
Great. So that was really a wonderful talk by Dr. Malay Shukla Saab on this very important topic. So, of course, tomorrow is the last day of our 21 days training course. And we have, the last but not the least theme, which is very important, about the policy interventions, a way forward towards the sustainable future. And we have scheduled 2 lectures for tomorrow by, very senior IFS officers. One start at 10:30 and will be about the forest and wildlife sector, policy, environment for sustainable management of biodiversity, followed by policy interventions in biodiversity conservation in India under international framework. So we want to give you a thorough, approach of the country-specific laws, legislations, programs in the biodiversity and environmental sector, and then an overall view of the world and how the cross-cutting themes are overlapping and how the Indian programs are, aligned with the global perspective. And 2 very senior IFS personalities will be delivering these 2 talks. In the second half, we have scheduled the valedictory session. We will send you the program and as well the schedule by today evening. Coming to the feedback by the participants, we have received the responses and we have lined up. I'm naming the participants: Dr. Shyama Parveen, Dr. Mohammad Maqbool, Dr. Brajesh Kumar, Dr. Ashok Gehlot, Dr. Aanchal Sharma. Apart from these 5 speakers for tomorrow for the feedback, we would welcome more participants to join for the feedback particularly. Of course, you all will be joining. Of course, a few are missing even today. That was the second last day. I hope tomorrow we will be having the full house. But if any of you like to give, your valuable feedback and views, are most welcome for tomorrow's valedictory session. And you may also, either text us or send us the, reply to that email which we have sent you. So with these words, thank you so much for your participation, and we look forward to your more enthusiastic and interactive participation tomorrow in the first half and in the post-lunch for the valedictory session.